Okay, today we're going to talk about cancer immunology and immunotherapy. It's a new area that's really, really hot, and a lot of people are interested. And my group is also devoting a lot of effort to develop methods in this area. So, if you look at the cancer mutations, if you remember some of the earlier lectures on this, we see that there are cancers that have less mutation. Usually these appear in pediatric tumors or tumors that face inside, for example, blood, brain, or prostate. Whereas there are cancers that appear in older patients, especially if they face outside, such as lung and melanoma. They have a lot more mutations. And in these tumors, once the tumor develops, even if you can find a drug that target the cancer, these are usually very difficult to treat because they have many other mutations and the DNA is not very stable. And so they can very easily evolve resistance against the drug. And so, for precision medicine, the one goal was to use really one targeted drug that specifically target the cancer mutation to cure the cancer and spare the normal cells. Unfortunately, if we use only single agent to treat the cancer that has many mutations, it's like trying to use a single policeman to clear up this crazy traffic jam. And so one very exciting discovery or development in the cancer world is the discovery of the cancer immunotherapy, or the development of cancer immunotherapy. In 2013, this is rated as the breakthrough of the year. And in 2018, the two early developers of cancer immunotherapy were awarded the Nobel Prize of Physiology and Medicine for their pioneering work in developing cancer immunotherapy. And so let's just look at the there are actually many different types of immunotherapy, but in order to understand that, first let's understand cancer immunology. So one question you ask people is, would tumor grow that's growing in one individual necessarily growing in another individual? So you can try this, in mouse. We know that very often we grow human tumors in mouse, or mouse tumors in another mouse. In fact, in cancer research, unfortunately, earlier in the last century, there are people who, they didn't know enough science. They try to grow one tumor in another person's tumor. Another person. And so the question is, would tumor grow in another individual? In most of the cases, they probably won't, because we have immunity. So the idea is, if we have normal tissues, initially, if there are some cancer cells, whether it's coming from the individual themselves or it's coming from external, very often the immune system can recognize this as foreign and reject it. This happens with organ transplantation. It could be that the host can reject if you don't eliminate or if there is no good matching, the receptor or the patient who accepts the donated organ might be rejecting the donated organs, right? This is because of our immune system recognizing something as foreign. And so very often when cancer cells really starts, our immune system is able to recognize this and eliminate this. Unfortunately, so these are usually highly immunogenic, transform the cells. They create a very strong immune response. But as we age, potentially people's immune system become weaker. And also, if the cancer is allowed to persist in there over time, it will evolve other mechanisms. One way is for this cancer cell to become less immunogenic. And so then the immune system can't recognize this very well. In addition, the cancer cell might evolve other mechanisms, which even if the immune system can recognize it, it will not really react strongly to eliminate the cancer cell. And so at this point, it's more in equilibrium. But then when usually by the time the cancer is detected in the patient is because the cancer cell has already evolved a mechanism so that it escaped the immune response. Even though the immune system is there, either it's kind of much, much weaker or it's being suppressed by the cancer cell. And so in general, the reason we can grow a tumor in a mouse is very often because that mouse has a suppressed immune system. Then you can actually put a tumor in the mouse to grow very quickly, okay? And so one of the earliest immunotherapy is the active immunotherapy. This is by giving the patient cytokines. So cytokines are cell-secreted proteins. When they encounter foreign particles, they can stimulate cell growth, inflammation, differentiation, repair, and immune response. There are many different cytokines. And one very important one is IL-2. And this cytokine, if you see, it's being secreted by some immune cells, but you can also synthesize this IL-2 as a therapeutic protein that you can give to the patient. And it can activate T helper cells, can activate B cells, activate T cells, activate NK cells. Not only they proliferate, they can also have higher activity. And this has been approved by FDA for treating kidney cancer and melanoma. And they just generally boost the overall immune cells inside the patient body. It's not specific. All the T cells get activated, which could cause problems such as cytokine storms. The patient has such a huge immune response. They usually have fevers that does not subside. In some sense, the side effect of IL-2 is a symptom that's very similar to COVID-19 response for the very acute symptom patients. Okay, so that's kind of the active immunotherapy. Yes, the patient has a much stronger immune system boost, which can eliminate cancer, but the patient can also have very potentially dangerous immune responses. What really got the cancer field to be very excited about cancer immunotherapy is the therapeutic antibodies that are blocking the immune checkpoint genes. And that's the main discovery of James Allison and Hangzhou. And so the idea is all of our cells, actually normal cells, normally express or present all the degraded proteins onto the cell surface for our immune system to check. You can imagine our immune cells, such as T cells and B cells, as guards in the body. They have receptors on the cell surface to do this checking, to check all the proteins that are presented on our normal cells to see, okay, these are ourselves or not. Actually, when our immune system or our T cells and B cells are maturing, there's a process to eliminate all the T cells and B cells that recognize self. And so any T cells or B cells that circulate in our body are only there to recognize things that are not self, or at least have stronger response to things that are not self. And so you can see, the T cells are having this receptor. By the way, every T cell has a different receptor. They recognize something different. They might recognize some virus or some bacteria or some other things. So not self. The normal cells will present the presented or degraded proteins on the cell surface, and when it's all normal, it's okay. But if, say, a normal cell is being infected by the virus, the viral protein will also be presented on the cell surface. Then it can be recognized by the T cell as a bad sign, and the T cell will eliminate the host cell that carries the virus. But in this case, for cancer tumor, we know that tumors have mutations, and those mutations can also be presented onto the cell surface. And that can be recognized also by T cells. And so this actually happens in every day in our normal individual without cancer diagnosis, because DNA replication has a natural percentage of errors, there could be some mutations that happen, but if we have an intact immune system, those can be recognized quickly, and those cancer cells can be eliminated by the T cells. The elimination happens in two ways. The T cells can recognize the cancer cell, and once this happens, when the T cells are activated, they secrete some cytokines. These are like bombing, right? So, such as interferon gamma, which basically makes the nearby cancer cells die. In addition, the T cells that can really recognize the cancer cells, they are also attached to the cancer cell, have a porphyrin which puncture a hole on the cancer cell and then secretes additional killing signals such as granzyme through this hole to the cancer cell, and then start the cancer cell death program inside the cancer cell. And so these things happen in our normal body. Unfortunately, if our immune system become weaker, one possibility is actually with age, our T cell diversity decrease because T cells are matured in the thymus, and the thymus function pretty much go away, or at least majority of it goes away after puberty. And so if you monitor people's T cell in the blood for their different T cell receptors, you will see that the diversity of the T cells decrease with age, and this rate actually decrease faster in male compared to female. This probably explains why females develop, or the percentage of patients with cancer are fewer in female than male. It's recently there are some hypotheses that or at least some projections that cancer will affect 1 in 3 in male, but 1 in 4 in female. And probably also as people age, live longer, this rate could be 1 in 2 in male and 1 in 3 in female. This is potentially because the male T cell receptor diversity decrease. So if a T cell that's supposed to recognize the cancer mutation disappears in your body, then the cancer cell can grow with that mutation. In addition, the cancer cell can learn to adapt the T cell response, and so they can evolve to have, or they can evolve to express some ligand, in this case PD-L1 or other ligand, which can interact with receptors on the T cells. So for example, if PD-L1, you can imagine it's like a little ligand which can interact, it's like an alcohol that interact with the receptor on the T cells. And so even though T cell initially use the T cell receptor recognizing the cancer cell as foreign, then this ligand will actually suppress the T cell from getting activated. And so the T cells can get really, dysfunctional or drunk in some sense and not do the killing job. And so the current immune checkpoint antibodies are these antibodies that can block the PD-1 or block PD-L1 or block the CTLA-4 so that the T cells can be activated. So basically they stop the ligand from interacting with the receptor. So PD-1 can no longer suppress T cell activation. And then it's like you pour ice water on a drunk soldier, right, or a drunk guard, and they wake up quickly and they say, wow, what happened? And then maybe they will be back on the fighting spirit again, and through the receptor they very quickly recognize the cancer cell as foreign and then can eliminate the cancer. And that's how the checkpoint antibodies work. So basically checkpoint blockade antibodies can activate T cells and also boost the immune system, but the T cells, after they are treated, they don't all activate. Only the ones that can recognize the cancer mutations will get activated. So compared to a general IL-2 treatment, checkpoint antibodies are activating the subset of those T cells that really are killing the cancer cells, are recognizing the cancer mutations. That's why it has much less side effect and it's kind of desirable. And those actually are creating long-term cures, and patients who can be completely cured from cancer and be tumor-free for many, many years, which is really quite exciting. Yeah, sorry, here's a tumor eliminated. So in addition to the checkpoint inhibitors, recently there are also development of CAR Ts. And so the idea is that the CAR T is developed from any individual cancer patient. You take their T cells. So by the way, these are not T cell cancers. These are, usually B cell cancers. So if we can get the T cells from the cancer patient directly, and then use engineering to engineer the T cells to carry a receptor. So on the T cell surface, these are, and so there's an antibody on the cell surface which can uniquely recognize the cancer cells. And then once it binds to the cancer cell or get activated, the internal T cell will start the killing mechanism to start kill the cancer cells. And so if we can engineer such T cells, then in the petri dish or whatever in the cell factory, you can treat these T cells with IL-2 to create an army, basically a special T cell troop, right? And you can clone many copies of this, and then you infuse the T cells into the patient. And then the T cells will circulate in the body, and anytime it sees the cancer cell with this receptor, the T cell will get activated to kill the cancer cells. And this has been really successful in treating B cell related tumors. And because B cell has CD19 and CD20 on the cell surface, which has good antibodies that uniquely recognize them. And so this can be used to treat B cell lymphoma or B cell leukemia. You do kill your good B cells as well, but hopefully your bone marrow can later on recreate the B cells. But the CAR T is like a one dose. You inject this dose. So basically you take out the T cells from the patient, engineer the CAR and amplify this T cell to 10 to the 10th. And then you do one shot injection to the patient. And hopefully that will wipe out enough of the cancer, or will wipe out the cancer cell. And then later on, hopefully the new B cells generated will be the correct B cells and the patient will be cured. And this one shot costs about $500,000, and you just pray that it will work. But there are trials, and these patients have been cured, and they work on mostly hematologic tumors because of the CD19, CD20 CAR works very well. There are challenges for solid tumor CAR T if you want to use CAR T for, say, lung cancer or breast cancer. There are a number of challenges. The first is we don't know what antibody are specifically recognizing cancer cells and are sparing the normal cells. It has to be really specific. And the second is because B cells, and these are usually circulating, so you just get the T cells, inject them in the blood, it will work. But for solid tumor CAR Ts, we need to make sure that T cells can actually get into the tumors. When they circulate in the body, they can really find the cancer cell and enter the tumor. And also, once the T cells recognize the tumor, it could be that the tumor can also quickly evolve PD-L1 to make the T cell dysfunctional. So how to maintain the CAR T cell activity to really do the killing job at the, all the distal solid tumor sites, that's also a challenge. The next immunotherapy is, so it's similar to the CAR T, is like a adoptive T cell transfer. Again, we need to, so in this case, if we can isolate the tumor infiltrating lymphocyte, this is directly taking a tumor out and separates the cancer cells and the immune cells. And so in this case, the cancer cell, you can kind of put them into little wells actually, for the immune cells, you can grow them into single cell clones. So you can see here, or a single or a little cluster of immune cells. And then you ask which of those immune cells really can recognize the cancer cells and secrete cytokine. That would mean this is a immune cell that uniquely recognizes the cancer cell. And when you see that, that's the clone you want to use IL-2 to proliferate a lot. And then again, you could externally create a mixture of immune cells. They all recognize either the same or slightly different tumor antigens. Then you inject this back into the patient, and hopefully they will kill the cancer cells in the body. And this has been done in both solid tumor and hematologic tumors, right? So, so this rely on our ability to recognize unique T cells that can recognize cancer cell antigens and secrete cytokines that kill the cancer cell. And then you externally amplify those and create a pool of this and inject back to the patient. Right? So in this case, we don't necessarily know what is the cancer antigen that this T cell is recognizing. But later on, there are also studies doing this. So after the cancer cell, the tumor are excised from the patient, we can separately do mutation profiling using whole exome sequencing from the tumor, but also look at, grow the immune cells. So in this case, after we do mutation profile from whole exome sequencing, the scientists can predict the immunogenic mutation, which mutation are likely to create an immune response. And in this case, all the mutations, you don't need to create the full gene. They create kind of a artificial gene cassette where each little piece is one mutation from the original gene. So you only need to create a little peptide of, say, 10 to 15 amino acid or little peptides where the mutation happens in the middle. So this could be the mutation coming from like BRAF gene. This could be the mutation from another one, like a recurrent mutation from the tumor, and so or a driver mutation from the tumor that's immunogenic, right? So if you can predict which of those mutations are immunogenic, we can put them in these little mini gene cassettes, and then engineer the dendritic cells in the patient to express the collection of these little mutations, and then co-culture the dendritic cell with the T cell. So basically the dendritic cell will present these mutations, and the T cells that recognize those mutations will get activated. And those, you can again activate those T cells and proliferate a big army of 10 to the 10s of T cells and inject them back into the patient. And those we can train the army to specifically recognize the cancer mutations, right? So those have also been done. There are also cancer vaccines, and most of the vaccines that people talk about nowadays, like flu vaccine, are prophylactic. You let the patient see the bad guys before. These are usually dead for like a smallpox or flu. These are some dead proteins on the virus or the bacteria, so that your body can generate the immune system. Unfortunately, for cancer, people haven't done any prophylactic vaccines. These are mostly done as therapeutic vaccine. The patient has already been diagnosed with cancer, and we can isolate the tumor, figure out which peptides represent some tumor antigens, so you can kind of mush up the tumor. And then train the dendritic cells. So in this case, the dendritic cell can present the tumor lysate. So for example, so dendritic cells are professional antigen presenting cells. They just chew, they eat up other cells and they present the chewed up material onto the cell surface. And so you can put the tumor lysate with the dendritic cell, and then they would present this onto the cell surface, and you inject just those dendritic cells back to the patient. Or we can create peptides that are representing the tumor mutations, and let dendritic cell present those onto the cell surface. There also work using viral vectors. This way, the viral vectors can be directly injected into the patient. The viral vector can carry a tumor mutation inside, and then they are injected into the patient. And interestingly, these viral vectors very often like to go to the location where the cell is proliferating a lot, because they want to kind of also hijack the system to grow, right? So they like to go to the cells that are growing a lot. And so they very often home to tumor site, and because they also present the tumor peptide, that they carry the tumor peptide payload, then they can also be recognized, hopefully, by our immune system better. And so there are these oncolytic viruses. They preferentially infect and lyse the cancer cells, and also they can stimulate the immune response. Okay, so these are other ways that people can use immunotherapy. And another recently very exciting way of immunotherapy is personalized new antigen vaccine. And so basically, if a patient is diagnosed with cancer, you can take the tumor out and do the mutation profiling, figure out which mutation might be presented or immunogenic, and take those immunogenic mutations. You can make a tube of those immunogenic mutations, whether as RNA or small peptides. So it's a collection of, say, 20 to 30 different potential immunogenic mutations, either as RNA or peptide, and then inject that into the patient. And then, for example, you can imagine if you give it a pool of peptides, and the circulating immune cells in our body will very quickly is like, here, let all the blood T cells see what are the bad guys look like. And those T cells, when they see the immunogenic peptide, they will proliferate, they will activate, and then they will also circulate in the body to identify the distal cancer cells and kill them, right? So, so there are also challenges, first of all, how to predict new antigens, how to ensure that those new antigens are presented somehow in your body so that the T cells can see it. It needs to be, if it's a peptide, it can be recognized, but if it's RNA, how do you make sure that they get expressed into a peptide to be recognized by the T cells, right? And also, we need to find the right mutations, and also make sure the T cells, if they are circulating, can go to the solid tumor sites to do the killing. And again, once they go to the solid tumor, we want to make sure they're not being suppressed by the PD-L1 or other checkpoint genes, right? So these are all challenges, but it's still, because of the patients that are treated and are responding well, showing very, very exciting, good responses, people are exploring all different ways. And that's why I think everybody in the cancer world are interested in cancer immunology now. And also, not only we can learn cancer immunology better, I think the efforts that are invested in this will help us understand infectious disease such as COVID-19, allergy, autoimmune problem, and also aging. A lot of other diseases are immune related. And so all of the work in cancer immunology are kind of relevant too. Okay, so that's on cancer, the different cancer immunotherapies. Questions on this? So the question is, yes, cancer immunotherapy is really interesting. What are the things that are interesting computational problems or genomic questions that are really worth investigating. First, I want to say that genomic and computational biology are always together. There is no genomics without computational biology, right? So we will show you a number of these questions in understanding cancer immunology, in understanding how this thing works in the tumor. And you can see how genomics is helping with this understanding and how computational biology is also helping in this. So the first question is, what mutations are in the tumors, right? So you can see tumors that are presenting mutations on the cell surface can be recognized by the T cells. So we have to know what mutations are there. So how do we get there? You can imagine we do mutation profiling, right? You call the mutations from the tumor compared to the normal, and you can clearly see some tumors have more mutation load than others. And in fact, the total mutation load turns out to be a very good indicator, or at least a repeatedly reported biomarker for which patient will respond to current immunotherapy. And so this is one paper that was published earlier in Science in melanoma patient. And you can see these are, each column is one patient. These are two different cohorts of patients that are treated with PD-1. And if you look at the response of the patient, PR is partial response, PD is progressive disease. So PR means that the tumor is, after the anti-PD-1 is shrinking by more than 30%. PD is the tumor is still continuing to grow. SD is stable disease, meaning that the tumor is not shrinking, but at least it's not growing. In this particular trial, we don't have CR patients, which are complete responders, meaning that after the drug, the tumor is gone. We can't even see it in pathology, which is, of course, the best case. But when, in this case, when the scientists look at which of those patients are likely to respond to tumor, to immunotherapy, on the left side, you can see these are more PR or stable disease patients. On the right side, these are more progressive disease patients. The biggest difference is their level of mutations. If they have more mutations, which is not surprising, you can imagine, very often the reason patients develop immunotherapy is because either their T cells are too dysfunctional because of PD-L1 or PD-1 or PD-L1, or because the T cells that are supposed to recognize the mutation are gone. They're dead. And so if a particular tumor has more mutations, there are more chances there are still some good T cells in the patient that can still recognize, maybe they don't recognize A mutation, but maybe they recognize B mutation. So this tumor is looking very foreign to our immune system, then they can be recognized easily, then they can be killed. And so mutation burden is a very good indicator of patient response to or checkpoint inhibitors, at least. And so that kind of, you go back to Dr. Gatti-Gazi's lecture on how to do tumor mutation profiles, how to call the mutations, and how to annotate those mutations, and understand which patient has more mutation burden, those patients are likely to respond to the current immunotherapy treatment. The second question that we are interested in are what mutations are presented onto the cancer cell surface, right? So this is related to antigen presentation. So we mentioned to you that all of our normal cells present degraded protein onto the surface, but different individuals actually present different things. This is because of HLA. So HLA and MHC, that is kind of interchangeable. One is the gene name, one is the protein complex name. HLA is the gene name, MHC is the protein complex. And so they are on the cell surface, and they present different proteins onto the cell surface. And so MHC binds and displays antigen onto the cell surface, which can stimulate immune response if the T cell receptor or B cell receptor or recognize what is being presented as foreign. And so if you look at our individuals, we mentioned that people usually have 99.99% DNA that's similar. The difference between individuals, one of the highest differences is coming from the MHC class 1 or class 2. It's very heterogeneous in population. In fact, if we want to do organ transplant or bone marrow transplant, the very important thing is to match the HLA type. For example, in HLA, we have HLA-A, HLA-B, HLA BRD1, and we also have one copy from our mom, the other copy from our dad. So there are 6 different alleles, say from a patient versus a donor. If all 6 matched, it's usually considered a good match. The donated organ can be transplanted to the patient. Whereas if you have 5 out of 6 match, it's just less ideal. And the more mismatch between the donor and the patient, it's going to have more rejections to the donor organ, right? So, which means that basically, depending on the different HLAs, different peptides will be presented onto the cell surface. And if the patient, previous T cells when the patient is young, is only trained to recognize, some things, and then suddenly you have a donated organ, because of its own HLA is presenting different set of things, they will all look very foreign to our immune system. Then that will be a problem. In order to call the HLA, there are experimental tests that can be done in the lab using sequencing approaches, but recently people can also directly get this from DNA sequencing or RNA sequencing from the tumor DNA or RNA sequencing. And so this is an example. After you get the HLA reads, you can just map them into the genome. And even though these reads are actually the HLA between the different alleles from different individuals, they could also be highly similar. And so we need a very good coverage along that region to say, this is really the HLA-A2 or HLA-A1. So what are the most dominant clone? Because we would have one copy from mom and one copy from dad. So usually we need to call what are the 2 alleles from each individual. You can, in this case, you can combine the germline reads from the tumor reads, because theoretically our cancer cells and our normal cells should have the same HLA. Occasionally you would see that the reads that are coming from the tumor can have differences from the germline. So supposedly from the blood, we see these are the HLA, but then somehow in the cancer cells, the reads look different. Those could be because HLA can be mutated. And we, in the next slides, will show you why those mutations happen. So actually in tumors, HLA mutation is fairly frequent. And so what are the different ways things are presented? So normal cells all have MHC presentation. The different MHC really bind to different peptides. And also for MHC1, this is one complex. They present just self-antigens. All of our normal cells present the degraded proteins. Just whatever is inside the cell is being presented on the cell surface. MHC2 is for cross-presentation. These are for professional antigen-presenting cells, such as B cells, dendritic cells, and macrophages. They eat other cells or CD4 T cells. They, they eat, CD4 T cells is not a professional antigen-presenting cell. Anyway, so B cells, dendritic cells, and macrophage, they eat up other cells. They can then chew it up and then present it onto the surface for other T cells and B cells to recognize, right? So they present not only themselves, they also present whatever they eat, including things from the bacteria or very infected cells and cancer cells. So during the immune cell maturation, we mentioned that our T cells and B cells are being presented by all the things that are ourselves as normal. And if a T cell and B cell has a receptor that recognize normal, they will not even have a chance to be kept alive. They will be kind of killed directly in the thymus or in the bone marrow. They don't really get a chance to circulate the body. Otherwise, you will have autoimmune problems where the T cells and B cells are recognizing self and is attacking normal tissues and organs. That's not something we want to happen. And depending on the different HLA type, this is one example of this is a sequence logo which represents for example, the peptide that is being presented by HLA A0101. This is one type of HLA-A from some individual. So if you have the HLA A0101, they are likely to present some peptide that has some motifs. The middle part can be anything, but there are some bases that are more specific. So this is just one example of MHC presentation. And what happens in the cancer is, for example, if we want to see which mutations are presented on the cancer cell, we first need to know the HLA type, HLA like a class 1. And so there are actually about 2,500 different HLA alleles that are available. You know, you can imagine there's like European, Asians, or African American, African, you know, depending on the ratio, or sometimes even small ethnicity differences, their HLA type might be different, and they will be potentially presenting different sets of peptide onto the cell surface. And unfortunately, right now, only 70% of the HLA alleles are well characterized. And there are computational resources such, you know, NetMHC is one very, very widely used algorithm that everybody in the new antigen prediction world, they use this tool. And they have good HLA recognition models for many different alleles. So for example, you can see here is HLA A3001. It recognizes different sets of peptides sequence. They will present those onto the cell surface. But in another HLA 3002, you can see the motif is very, very different. They will present a different set of peptide onto the cell surface. And so, yeah, as we mentioned, currently the NetMHC for HLA A and B molecules, we have a pretty good prediction, but for the not so common HLA alleles or for HLA class 2, it's not present. It's the current computational model for those are not very good. And so basically, if a patient has a mutation, this mutation needs to be presented by a particular HLA type for that patient very strongly onto the cell surface. Otherwise, this mutation may not really be presented on the cell surface. Then it will not be visible to our T cells, right? So, and so that's very important. In addition, later on you will see that even antigen is being presented on the cell surface doesn't mean that it will necessarily generate an immune response, which is kind of tricky. Another question that's very cancer related is there is a tumor mutation bias due to MHC class 1 of the patient. Basically, there are many mutations that are occurring just because they are not being presented by the HLA type. So for example, if a mutation is initially happening by chance in the tumor, if it gets presented on the cell surface, then our immune system can recognize this very quickly and eliminate the tumor. A subset of those tumors were able to develop because they could have a driver mutation or a passenger mutation, which does not get presented onto the cell surface. Because the HLA type of this patient never present this type of peptides. You know, you can imagine, if this is a mutation that happens on this base, on this K, which is normally should be presented on the HLA-A 3001, but this patient only have a 3002, then this will not really be presented onto the cell surface. Then you won't really see it, or at least the immune system will not be able to detect it. And so there is a Cell paper in 2017. They look at all the mutations in the patient tumor and all the HLA type for the patient, and they see that in general, the mutations that you see in specific tumor are enriched in those mutations that are not presented by the patient. In addition, sometimes, as we mentioned, HLA gene itself can be mutated. And a lot of those mutations happen just so that the mutations in the patient are no longer being presented by the HLA, because if the HLA is not working, then it won't really be presented on the cell surface. So you can see here, antigen presentation is a very important thing. We not only need to know the mutation, we need to know the HLA type, then we will know which kind of mutations will be presented onto the cell surface and be recognized by our immune system. And those are all currently done with genomics and bioinformatics. Okay, questions? The next question we ask are, what immune cells are infiltrating the tumors? So in projects such as TCGA, when they were developed, there are pathologists looking at the tumor to ensure that the tumor has over 50% purity. Because when you do DNA or RNA sequencing, you want to make sure you are really sequencing the tumor and not really sequencing a lot of the normal. And this is in the early days of TCGA. You think that tumor with high purity is really, really important. But later on, scientists realized that a lot of these normal cells are not necessarily, for example, you don't want to sequence normal breast epithelial cell. You want to sequence the malignant epithelial cells. But a lot of the normal cells here are not necessarily normal epithelial cells. They could be immune cells, T cells, B cells, dendritic cells, fibroblasts in the tumor, which are actually quite important. And so especially in the tumors, the very good immune cells that can kill the cancer cells are T cells and NK cells that are key factors for patient survival and immunotherapy efficacy. And so pathologists are now learning to stain for this immune cell infiltration. They are also called tumor infiltrating lymphocytes. And so you can see this is one tumor where the tumor immune infiltration is low. This is less than 30%. This is, this is staining for immune cells. And this is what we call immune hot tumors, or tumors with abundant infiltrating lymphocytes. And so this is from pathology, but recently there are also algorithms that can predict immune cell infiltration in the tumors based on RNA sequencing data. So there are two different approaches for this. One is a gene set enrichment type of approach. Basically, if we know what genes are uniquely expressed in T cells, what genes are uniquely expressed in B cells, what genes are uniquely expressed in macrophage, we can create those gene signatures. We say, okay, these 50 genes, these 20 genes, we create a signature, and we use this signature to evaluate each tumor to say, in this tumor bulk RNA-seq, do I see a high signature for B cells? Do I see a high signature for T cells? And so that will kind of give us kind of a relative estimation of immune cell signatures across different tumor types or tumor samples. And so this is one approach. They are using kind of a gene set enrichment analysis to evaluate the immune cell infiltration. The other type of algorithm is an immune deconvolution approach. So if previously people have done a sorted immune cell expression profile, we might have information of how dendritic cells, it is like a sorted dendritic cell, we have their genome-wide expression. And this is kind of a, say, T cells or maybe B cells, you can get their expression. And you can either use all the genes, or usually people pick out still, maybe tens or hundreds of genes that are kind of uniquely expressed in those immune cells, and then do immune deconvolution. So basically, you think that the final tumor expression is the different immune cells that are mixed at different levels, which will give us the final expression level of all these genes. And so the immune deconvolution method is to ask, okay, if T cells express these genes at this level, dendritic cell express these genes at this level, macrophage express these genes at different level, what level, what infiltration level can I estimate? This fraction or the estimation of the abundance is what you try to do. You estimate this in order so that at this proportion of T cells, this proportion of dendritic cell, or this proportion of macrophage, you will have the final expression that's observed on the tumor. And so this is a deconvolution type of approach. There are also many algorithms developed. And so far, analyzing a lot of different TCGA tumors, it seems that immune cell infiltration really have very strong influence on patient survival. For example, if the tumor has overall very high level of macrophage infiltration, in here, each column is a cancer type, and each row is one major immune cell infiltration level. And the little square indicates the survival death risk of the patient. And red means bad, blue means good. And if the square is big, it means the p-value is more significant. And so you can see here across many cancer types, overall, this is very red, even though only a subset of those are statistically significant. But what it says is that when the tumor have overall a higher macrophage infiltration, patients usually don't do so well. But if they have a very high level of CD8 T cells, you can see in this row, it's mostly blue. If the tumor has overall higher CD8 T cell infiltration, they usually do better. And so that's kind of a useful thing, based on the tumor, whether you can check on the tissue slides or based on the RNA sequencing, you can already make some guesses of how well the patient will survive. Recently, we developed a computational algorithm called Timer. We have our own algorithm, which is a deconvolution method, but we also incorporated 5 other immune deconvolution type of algorithms together to evaluate all of TCGA. So for example, if you want to ask how is antigen presentation associated with CD8 T cell infiltration, so the Timer algorithm has already looked at the expression level of the genes and also the immune infiltration of the different tumors in TCGA. And so here now each row is one cancer type and each column is a immune infiltration estimation methods. For example, there's a Timer estimate, there's EPIC estimate, and Excel is a different algorithm, QuantiSeq is a different algorithm, but you can see red means positive correlation and blue means negative correlation. So from this, you can see overall, when the tumor has higher B2M gene expression, the tumor overall have higher CD8 T cell infiltration because you see the expression of the gene and the CD8 T cell infiltration are positively correlated in many cancer types, regardless of what algorithm we're using to make the prediction. And so this kind of give you some idea what genes are really important in helping in T cell infiltrating into the tumors, right? So this is a useful thing. Recently, there are also single cell techniques such as single cell RNA-seq. You can also use this to estimate the immune infiltration. And so if you remember from the single cell RNA-seq part of the lecture, in the tumors, there might be malignant cells, but there also can be T cells and B cells, and you can compare this. So there are some important factors to know. First, it's okay to compare the same population across different samples. Say, if you want to ask, overall, when we look at CD8 T cells, you can ask which samples have higher CD8 T cells. But then we have to be a little bit careful when we compare different cell populations in the same sample. For example, you can't really compare the absolute level of neutrophils versus T cells, because for some reason, neutrophil is very difficult to be prepared into a single cell RNA-seq reaction. A lot of neutrophils, as you're preparing, they're dying, so it doesn't really get into the single cell suspension. And so the relative level of different, especially totally different immune cell types, we have to be cautious about what we conclude, because they may not have similar level of, or they may not be easily made into the single cell sample preparation. Also, it is good to compare the differential expression of different cell clusters in the same sample. So for example, if in the same tumor, I see kind of a progenitor T cell and a exhausted T cell, or dysfunctional T cell are those drunk T cells that are no longer killing the cancer cells. You can compare the expression of those two cell types in the same single cell data. But when you compare the differential expression of the same cell cluster across different samples, you need to be careful, because some people will just, process, two different tumors, say one is responding to immunotherapy and one is not responding to immunotherapy. You do single cell RNA-seq. When you overlay those data together, you might see differential expression. And because there are hundreds of cells in that cluster, you might even have a very significant p-value. But the reality is you only have one patient in group 1 and another patient in group 2. It's never going to give you a confident enough readout. And so be very, very careful when you are comparing, the differential expression of single cell across different samples. So make sure you still need to have biological replicates to really call differential expression, and also to call the differential cell populations. Okay, questions? So we mentioned that on the cancer side, there are mutations in the tumor, and through MHC or HLA, they are presented onto the cell surface. And then in the tumor, we also know that the T cell has to infiltrate. If the T cells don't really get into the tumor, then they certainly cannot see mutations. But even after they get into the tumor, in order to recognize the tumor as foreign, we need this thing called a T cell receptor to really recognize or distinguish self from non-self. And so how is that done? Every human being, like every individual, we have only 2 copies of the chromosome. But in order to really recognize self with non-self, the TCR has to be able to recognize millions of different things, especially to recognize like viral bacteria infection or some fungus, these type of pathogens. How is that done? This is through a process called VDJ recombination. So basically, in the multicellular species with kind of a T cells, you will see the situation like this. In the T cells, there is a region of the genome which code for a T cell receptor. It's a gene fragment, which means it's not like normally as you would think as one gene, is a fragment. And so in here, there are many V fragments. The orange is a D fragment and blue is the J fragment. So in here, you can see for T cells, all the V's are next to each other. And then there is one D and a whole bunch of J, another D, a whole bunch of J, another D and a whole bunch of J. And so in VDJ recombination, what happens is that the cell will randomly pick 1V, 1D, and 1J and stitch them together. Actually, the DJ recombination happen first, and then is the VD recombination, and you stitch them together. You can see here, if the V that's picked is the second one, in this DNA, the ones before this will still be present, but the one after that will be deleted from the DNA. So for example, if you pick this V and this D, everything in between will be deleted from the DNA. And so this two will be stitched together on the DNA. And if you pick this D and this J, everything in between will be deleted, but the J after that, you can see here, the J after that will be still kept. And so at the very end, only the VDJ that's really close to each other will have a chance to be made into a functional T cell receptor. And the things outside, this V or this J are not really used. So it's only middle one that's important. So there are ways to do the TCR sequencing in usually you do this in the blood, or you can also do this in the tumor. And this is using a multiplex PCR reaction to amplify out all the possible VDJ pairs. And so basically you have one V in here, one like PCR primer on this, all the V and all the J. And so that will amplify out all the VDJ pairs. And after that, there needs to be a site selection because the non-productive VJ will also get amplified, but you don't want those. You just want the correct size VDJ that's closest to each other to be amplified out. And the final result, what you get is you say, okay, this VDJ sequence is this abundant. Another VDJ sequence is at this abundance, and another VDJ is at this abundance. One thing we want to mention that VDJ recombination, not only the combination creates the diversity, but also in order for this recombination to happen, you cut the DNA in here, you cut the DNA in here, and you try to stitch these two together. And this stitching process is never perfectly accurate. And so in the stitching location, there could be small insertion, deletion, and mutations in there. And that creates additional diversity into the TCR. And so every individual, even though you only have, just one copy of the VDJ segment from mom and another VDJ segment from dad, from this VDJ recombination, not only from the combination, but also from the end, creating the small indels and insertions, every one of us is able to create, in theory, millions of different TCRs that can recognize different antigens in the sample. And so when you do a TCR sequencing, for example, in the blood, what are some of the interesting things we see? One thing we see is that because thymus is what is used to train the TCR to call what is self and non-self, all the TCR that recognize self will be killed in the thymus, whereas those that are not recognizing self are allowed to circulate in the body in order to recognize outside pathogen. After puberty, our human thymus will go away. And so most of what we have in our body are the T cells that were trained during puberty. If they have a chance to be amplified through stimulation, say it's seen some mutations or seen some pathogens, it amplified, it create multiple copies of itself. Then they will live longer. But if a T cell has been around for a long time, there's never a stimulation, over time it will die. And that's why if you look at the T cell diversity, some very interesting observation is as people age, because our thymus pretty much lose function, the overall T cell diversity in the body decreases. And also male decrease faster than female. And so this probably explains why male have more cancer incidence than female and have shorter life expectancy. So female has kind of a slightly better, I guess, uh, longer maintained immune health. And so overall TCR diversity, if you look at age and gender, there's actually a pretty good curve. And so you can imagine if you just monitor people's TCR diversity, you can say, okay, normally male at 50 years old should have this high T cell diversity, but then if an individual has a very low T cell diversity, they have to be careful because this indicates that the patient can, or this individual, not necessarily a patient, can have mutation, but if the T cells that's supposed to recognize that mutation is gone, there might be cancer development, and the individual's T cell won't be there to really recognize it. And so this is one study we did published in 2020, where you look at the healthy donors. You can see that they have very high T cell diversity, but for Hodgkin's lymphoma, this is actually a kind of cancer where it happens fairly early in young adults. Usually even teenagers or 20, 30-year-old people can have Hodgkin's lymphoma. And you can see the newly diagnosed patient have much low level of T cell diversity. So even though they are only 30 years old, their T cell diversity is like a very old man. And so also if we look at those relapsed patients which have just failed many different drug treatment, they are kind of metastatic, and hopefully these patients, their T cell diversity could be even lower. And interestingly, when we look at how these patients respond to immunotherapy, and so this is the newly diagnosed, and if we divide up this relapse group later on to give them immunotherapy, one thing we notice is that those patients that actually responded better, they had a complete response, meaning that the tumor is totally gone, they have still overall higher T cell diversity. Whereas those patients that have, a partial response or even progressive disease meaning the patient don't respond very well, they have a lower overall T cell diversity. And so basically, you need to have enough different T cells to recognize mutations for you to have the chance of going into the tumor and say, that's the bad guy. And so in your body, in your blood, T cell diversity is definitely a good thing. And interestingly, when we look at the patient after, so this is before the patient were treated. When we compare the before treatment to after treatment, you can see those complete responders, when you treat with immunotherapy like PD-1, their overall T cell diversity increases. It's like, you make sure that the T cell that previously only had one copy maybe now had a chance to make a doublet of it, like make a clone of itself. And that's actually a good thing to ensure that, you don't, when you have only one copy left, you don't want this T cell to die, but, with immunotherapy, okay, now you created another copy of it, right? So that can increase the T cell diversity. But interestingly, for the partial responders, and especially for the progressive disease, you don't really see this type of T cell diversity increase. And so we can see that T cell diversity is actually a very good measure of immune health and also can be predictive of immunotherapy response. In recent years, there are technologies such as 10x genomics that allow people to do not only single cell RNA-seq, but in the T cells, you can specifically amplify the VDJ, and based on the cell barcode, you can match the TCR T cell receptor to the cell that you have the single cell expression. And so this is also a study published last year where they look at the TCR clones and ask which T cells are clonal expanded. By clonal expansion, we mean that multiple T cells have the same TCR clone, identical TCR clone. That means there's a clonal expansion. The T cell is making clones of itself because the T cell is identical. And then for those expanded clones, you can ask, do their expression look different from the non-expanded clones? Because in the tumor, or interestingly, with immunotherapy, you want the T cells that recognize the bad guy, the mutations in the cancers, to have the chance to expand. So in the tumors, clonal expansion is a good thing. You want specific clones to be activated and start doing the killing job. Right, so so TCR sequencing in the single cell is very important to help you figure out, oh, those expanded clones actually have different expression from those non-expanded clones, which are just parking lots there, they're just waiting. And so, of course, we still need to discover what those tumor, what those TCR really recognizing, what antigens do they recognize. And so this is one study that's published a couple years ago, where they look at TCRs that specifically recognize KRAS mutations in melanoma. So melanoma cancers have very frequent KRAS and BRAF mutations. These are inside the cell. The KRAS and BRAF are cell signaling molecules inside the cell. But if they are mutated, their protein product, when it's degraded, can have a chance to be presented onto the cell surface. And so an antigen presented KRAS mutation can be recognized by the T cell as kind of the antigen to create an immune response. And so you can imagine if you have an antibody that recognizes the presented mutation, or if you have a TCR that recognizes the MHC presented antigen, you can engineer those T cells to specifically kill the cancer cells. And so this study actually was from Steve Rosenberg, looking at a T cell receptor that can recognize a presented KRAS mutation as a cancer therapy. So you can create many, many copies of those T cells and then infuse to the patient. And recently, there's another study from Bert Vogelstein, where they look at a CAR, which means that there's an antibody which recognizes MHC presented P53 mutations. And then those T cells will only go to the tumors with those P53 mutation. And so basically CAR is the receptor is the antibody, or the protein that recognize the tumor antigen, whereas TCR, you are using kind of the TCR to recognize MHC presented mutations. Yeah, so you can see here, TCR is really important to understand the overall immune health, to understand which T cells get activated in tumor and have clonal expansion. And you can even create a TCR specific T cells as kind of a special weapon and amplify them and infuse them into the patient as a type of immunotherapy. Okay, so that's good for today.